So welcome to the Master of Marine and Environmental Science capstone webinar at the University of the Virgin Islands. My name is Lorraine Buckley, and I am a professor of biology and the director of the MMES program. As program director, I'm one of 6 faculty that advised this year's capstone project. First, I'll tell you a bit about the capstone project. Each year in their second semester, the MMES students are challenged to develop a major publishable project that is hypothesis driven and that has a relevance to the Virgin Islands territory. The capstone reporting at the end of their project includes information for resource managers, a community outreach component, a manuscript for publication, and a report for science professionals, which this year is today's webinar. The capstone is graded, and that grade contributes to each of the 4 core courses that the students take in the spring semester of their first year. This year's capstone project investigates the role of herbivory on coral reef recovery, and it uses databases that are shared on the Marine Biodiversity Observation Network, or MBON. MBON was developed to share knowledge and methods to evaluate changes in the biodiversity of the oceans on a global scale. Locally, the nonprofit called OCOVI, or Ocean and Coastal Observing Virgin Islands, and its regional group CARICOOS, and the National Integrated Ocean Observation System, or IOOS, are committed to MBON, and OCOVI contributes local data to this global network. As a founding officer on the board of directors of OCOVI, I'm particularly excited that the 2020 cohort of the MMES program developed a project using MBON data. In planning their capstone, the 2020 cohort made the best of the pandemic limitations by using MBON data from the Virgin Islands and Puerto Rico to investigate their questions about herbivory on coral reef recovery. To my knowledge, this is the first use of the local MBON data to answer broad ecological questions relevant to these islands. So thank you for joining us today. And it is now my pleasure and honor to introduce the fabulous 2020 MMES cohort, the first cohort to complete their entire core curriculum, 18 graduate credit hours, entirely online. Representing her cohort is Allison Holbrook. Allison?
Thank you so much, Lori, for that wonderful introduction, and good afternoon, everyone. I'm going to take a minute to get my screen set up here. I would just also like to make a note to please have your volume turned up on your computer. It is raining on the north side, and although my volume is turned up on the computer, it might be a little bit loud should the rain come in and give us some needed water for our cisterns.
Good afternoon, everyone.
Okay, so before we jump in, I just wanted to say thank you again so much for attending, and I'm gonna just go right into the show.
Good afternoon, everyone, and thank you for joining today's webinar. For your viewing pleasure, this webinar has been pre-recorded. If you have questions during the webinar, please type them into the Q&A box on your screen, and questions will be addressed at the end. Survey links regarding today's event will be shared at the end as well, and we thank you so much in advance for your feedback. Have an afterthought? You can email your questions to us up to 2 weeks after this webinar. Questions received after the 2-week deadline will not be answered, and this email will reappear again at the end of the show. All pictures are from canva.com unless otherwise noted. And we are your hosts from the 2020 MMES cohort at the University of the Virgin Islands. I'm Allison.
I'm Jenna.
And I'm Tasha. Although you will only be seeing our 3 faces during the presentation, there are many wonderful brains behind this effort. We would like to give a quick shout out and acknowledge the hard work, dedication, and creative energy that has gone into this presentation from our cohort members. Thank you. Now sit back, relax, and enjoy the show. All right, welcome to our capstone presentation. This semester, we went fishing for answers regarding the role of herbivory on coral reef recovery in the US Virgin Islands and Puerto Rico. Coral reefs are regarded as one of the world's most productive ecosystems, home to a variety of organisms. Each organism plays an important role in the reef, and the interactions of these organisms contribute to the functioning within reef systems. One key organism in a reef community is coral, and here in the Caribbean, we have around 70 species of corals. Besides providing aesthetic value for land dwellers such as ourselves, corals provide a variety of important services. Corals actually build the reef by secreting calcium carbonate beneath their bodies as they grow. They protect our coastlines from storm surges and swell, and they provide habitat and protection for ecologically and economically important fish species. Now, coral species vary by depth, creating different niches along the depth gradient. In the Caribbean, we have shallow reefs as well as mesophotic reefs. Shallow reefs are in the nearshore environment, and they're less than 30 meters in depth, while mesophotic reefs are found at depths of 30 meters or more. Different species of corals can be found in these different depths based on their ability to survive in different conditions. Corals in the shallows depend on lots of light and include a variety of species, such as branching coral, one of the major reef-building corals. The word mesophotic actually means meso, middle, and photic, light. Mesophotic corals are adapted to lower light conditions and cooler temperatures. Instead of having dynamic branching forms, mesophotic corals tend to be more round and even flat like plates. These forms increase the surface area that is exposed to potential light so that corals can photosynthesize. But no matter the depth, the ability of coral to live and grow influences reef recovery after disturbances. One factor of this recovery is coral reef resilience. Reef resilience includes the capacity of a reef system to absorb disturbances by maintaining functionality and returning to a pre-disturbance state after a stressor. However, competition between corals and macroalgae can lead to further stress. With high recruitment and fast growth rates, macroalgae are their fierce competitors that are quick to take up space that the corals would otherwise use. So how can corals keep up? To compete with their macroalgae neighbors, corals need to stay healthy. They need increased fitness in order to allocate energy to fighting disease, feeding, and reproduction. Overall, perpetual stress diminishes a coral's ability to compete and to recover from disturbance. Luckily, corals can have some stress-reducing help from herbivores. Herbivores are animals that feed on plants, and in reef systems, herbivores can include invertebrates like sea urchins, megafauna like sea turtles, and a great number of fish species. Each herbivore provides a different service and function to coral reefs, and different kinds of herbivores can be found at different depths, just like corals. But one of the most important kinds of herbivores on coral reefs and the focus of our presentation today are parrotfish. Parrotfish belong to the family Scaridae, and in the Caribbean, there are two genuses. There's Scarus, which includes species like Scarus vetula, the queen parrotfish, and Scarus icerti, the striped parrotfish. And the other genus is Sparosoma, which includes Sparosoma viridae, the stoplight parrotfish, and Sparosoma rubripin, the yellowtail parrotfish. These fish provide a number of services to reef ecosystems, but perhaps most importantly, they manage algal growth on the reefs. Due to their varying mouth morphologies, parrotfish species forage on reefs in different ways. Parrotfish feeding methods can be subdivided into three functional groups. We have the scrapers, which are voracious feeders and scrape lots of algal material and preferentially forage on algal turfs. These include species like the midnight parrotfish and the rainbow parrotfish. We have grazers, which preferentially forage on macroalgae and forage at a lesser rate than scrapers. These include species like the yellowtail parrotfish and the red band parrotfish. And lastly, we have the bioeroders and excavators, which take large bites out of the substrate to consume plant material and often end up scarring corals during the process. Excavator species include the stoplight and the queen parrotfish. Now, parrotfish forage primarily in the epilithic algal matrix, or EAM.
which is the primary substrate in most reef systems that's comprised of detritus, sediments, and algal turfs. They exert top-down control and suppress the growth of algae that may otherwise outcompete corals, thus resulting in a healthy coral-dominated reef. Remember, we just talked about the different roles that parrotfish play on reefs. Well, just to reiterate, larger scrapers in the genus Scarus, like the rainbow, blue, and striped parrotfish, preferentially forage on algal turfs in the EAM, thereby suppressing the growth of macroalgae. Smaller grazers in the genus Sparosoma, like the yellowtail parrotfish, tend to prefer macroalgae. And lastly, some species like the queen and stoplight parrotfish are bioeroders, so they remove algae from the reefs, but in the process of foraging, they also remove some of the calcium carbonate coral skeleton. So what do these various roles mean for reefs? Well, it's not just the abundance of these herbivores that's important, it's also the functional diversity. And these two factors together play a pivotal role in maintaining coral reef health. But stressors, such as overfishing and sedimentation, can directly impact herbivore populations, resulting in significant declines of larger species, which then tend to be replaced by the smaller species. Now, you might be asking yourself why this is such a big deal, and the answer lies within grazing intensity and grazing pressure. Larger scarabs remove more algae and forage at higher rates than smaller Sparosomids. Thus, they are able to prevent macroalgae from overtaking coral reefs. But when those larger grazers are replaced by smaller ones, there's less grazing pressure and less grazing intensity. This allows for competitive macroalgae to become established and proliferate, outcompeting corals for space and thus reducing recruitment and survival. Although smaller species preferentially forage on this macroalgae, they are unable to graze at sufficient rates to control it. So what ends up happening is we see a phase shift from a coral-dominated to an algae-dominated community concurrent with this decline in ecologically important herbivores. As parrotfish graze the substrate, removing algae, they can have a positive effect of maintaining a coral-dominated community by driving the composition of algae through top-down processes, thus helping to provide room for coral recruitment.
As Jenna mentioned, a reef's ability to recover from disturbance depends on both the abundance and functional diversity of herbivores. Unfortunately, algal dominance is associated with declines in reef health. And the removal of these herbivores by fishing can facilitate this decline. Although many Caribbean regions have already experienced a shift towards an algal-dominated state, the role of grazing intensity in driving overall reef resilience is not well established. Globally, corals are threatened by a variety of stressors, including sea level rise, ocean acidification, and global warming. Here in the Caribbean, local stressors such as disease, poor water quality, including sedimentation and excess nutrient input, as well as overfishing are further stressing our reefs. Hughes et al. 2018 found that increases in the severity and frequency of disturbances have shortened recovery times for reefs. Less recovery time between disturbance events means just that, less time for recovery and consequently an increase in stress for corals. Given adequate time and favorable conditions, corals can recover. We had a unique opportunity to assess this relationship in our own backyard. To do this, we had to have a disturbance event as our starting point to evaluate recovery. Since 2000, there have been 2 acute disturbance events to Caribbean reefs. First, being a significant bleaching event in 2005, which was followed by a spike in coral disease due to the stress in 2006. Secondly, 2 major hurricanes tore through the Caribbean in 2017, Hurricanes Irma and Maria. Therefore, the data that have been collected between the years of 2007 and 2017 were chosen to evaluate recovery over time since these years were relatively disturbance-free. That brings us to our study, where we aimed to determine whether herbivore biomass drives the rate of coral reef recovery over an undisturbed interval following a major thermal stress event across Puerto Rico and the US Virgin Islands. Although our capstone is still a work in progress, our main hypothesis was that sites with higher herbivore biomass show faster rates of positive change in coral cover. It is important to note here that for our study, we use herbivore biomass as a proxy for grazing intensity. We opted to use data collected in Puerto Rico and the US Virgin Islands through 2 long-running coral reef monitoring programs in order to investigate the relationships between herbivores and reef recovery in 2 distinct island communities that are in close proximity. We used extensive data classifying reef characteristics and herbivore biomass collected through the Territorial Coral Reef Monitoring Program, or TCRIMP, in the US Virgin Islands, and the Puerto Rico Coral Reef Monitoring Program, or PRCRIMP, in Puerto Rico. Although our data come from TCRIMP and PRCRIMP, we will be referring to these data sets as the US Virgin Islands, or USVI, and Puerto Rico, respectively, for ease of communication. And just before diving in, we wanted to take a moment to recognize the organization that has been working to bridge the gap in data accessibility between large monitoring efforts around the world. The Marine Biodiversity Observation Network, or MBON, is a global initiative designed to integrate biological and environmental data from long-term monitoring programs into a shared database, enabling the scientific community to better describe the relationships between marine biodiversity and ecosystem function. Their website is included at the bottom of the page for your own perusal. This network strives to collect data on common methodologies and share information on a continental scale. Contributions from the Puerto Rico and USVI regions are associated with the NBON Pole-to-Pole program, which aims to extend the sharing of biological and environmental information along the Pacific and Atlantic coastlines of the Americas. Through NBON, we were able to access the Puerto Rico data for our use in this study. The US Virgin Islands data is not yet available and is currently being uploaded and will be available through the portal soon. The US Virgin Islands and Puerto Rico monitoring programs both collect information on fish species, as well as information on the benthic community diversity and health along transects at their respective sites. Both programs collect data with diver surveys or through the use of point count analysis of photos taken of the benthos. It is important to note here that the methods do vary slightly between the 2 studies, so some standardization was required across the board in order to make comparisons between the sites monitored by each program. We don't want to spend too much time going through the specifics though, so please feel free to pop your questions about methods of standardization into the Q&A box. Trying to analyze these huge data sets proved to both be a blessing and a curse, as we had so many variables that we can investigate. Here on this slide, you can see a few major ones, but our main goal for the analysis was to take each of these variables and look into their relationships, specifically with the rate of coral cover change over time at our sample sites. Before Tasha and I dive into the results, remember that our hypothesis was that sites with higher herbivore biomass show faster rates of positive change in coral cover. So what did we find? Well, our results don't scare us so far. We wanted to assess our primary variable first, which was the change in cover coral over time. Here, we represent the change in coral cover over the years through box plots. Keep in mind that these are representing the coral cover change over a period without any significant disturbance. Anything located to the right of the center line represents a positive change in coral cover, or you can think of this as reef growth. Anything located to the left of the center line represents a negative change in coral cover over time, or you could think of this as reef decline. All sites here are listed in order from shallow to deep, moving down the figure. And here we are also looking at all sites within the US Virgin Islands. You can see that several boxes hover around the center line, suggesting that these sites do not have any real decline or growth, but are stable throughout the years. There are also several boxes that are to the left of the line, suggesting that there have been a decline in coral coverage over the years since the 2005 thermal event. And then there are several boxes to the right of the line, indicating a growth in coral cover or possible reef recovery. So we can see that coral cover varied quite a bit among the sites in the US Virgin Islands, with no consistent trends during this disturbance-free period. Remember, this is important given that our hypothesis was that sites with higher herbivore biomass show faster rates of positive change in coral cover. We also evaluated the change in coral cover in Puerto Rico. Again, this figure represents the change in coral cover over the years assessed through box plots. Anything located to the right of the center line represents a positive change in coral cover, or again, reef growth. And anything located to the left of the center line represents a negative change in coral cover over time, or reef decline. All sites here are listed in the order from shallow to deep, moving down the figure. While the sites in Puerto Rico varied, the majority of sites lie to the right of the line, indicating a positive growth in coral coverage. However, the time frame in which we were able to evaluate the change in coral cover was on a more condensed scale, as not all years between 2007 and 2017 were consistently sampled. These changes could be potentially magnified. After looking at changes in coral cover, the next field we wanted to assess for our hypothesis was herbivore biomass. Here, we combined all herbivorous fish to examine how the average total herbivore biomass varied among sites in the US Virgin Islands. Again, the sites here are listed from shallow to deep, moving down the figure, and the boxes encompass the years sampled. On the x-axis, the average biomass in grams per meter squared increases as you move from left to right. From this figure, you can see that the average total herbivore biomass does vary by site, like that of the change in coral cover, but was generally between 25 and 50 grams per meter squared over sampled years at most sites. Furthermore, we see a possible slight trend towards a decrease in total herbivore biomass at depth increases.
After assessing the composition of the total herbivore biomass, we discovered that 63% of the total herbivore biomass in the US Virgin Islands was comprised of parrotfish. So we decided to isolate average parrotfish biomass over the sampled years by site. Same as the total herbivore biomass, we have arranged the sites by depth, and the biomass in grams per meter squared increases from left to right. Overall, average parrotfish biomass over the sampled years was generally between 0 and 40 grams per meter squared. On this figure, though, we don't really see that same trend of decreasing biomass with increasing depth. However, what we do see is an almost distinct separation of biomass between the sites less than 30 meters and the mesophotic reef sites. For the mesophotic reef sites, we see what looks to be a trend of decreasing biomass with increasing depth. But this trend does not look to be present in the sites above 30 meters. Now, we all know that you're wondering, how does this compare to the rest of the Caribbean? Well, we've got you covered. In a study by Chance et al. 2020, they found that the average parrotfish biomass was approximately 12 grams per meter squared in the Bahamas, approximately 19 grams per meter squared in Jamaica, and approximately 24 grams per meter squared in Turks and Caicos. Since we observed these patterns with the herbivore biomass in the US Virgin Islands, we were interested to see if they also existed in Puerto Rico. Same with the total herbivore biomass in the US Virgin Islands, we created this figure with sites listed in order of increasing depth, and the average biomass in grams per meter squared on the x-axis. Here we found that overall, the average total herbivore biomass across the sampled years was lower in Puerto Rico than in the US Virgin Islands, hovering between 0 and 25 grams per meter squared. Whereas mentioned before, the US Virgin Islands sites' average total herbivore biomass was generally between 25 and 50 grams per meter squared over the sampled years. Additionally, here we did not see the trend of total herbivore biomass decreasing with increasing depth. However, this data set did include fewer families of herbivores, as well as a narrower range of years. So these factors most likely played a role in the differences between average total herbivore biomass in Puerto Rico compared to the US Virgin Islands. Just as in the US Virgin Islands, we found that a large portion of the Puerto Rico herbivore community was dominated by parrotfish biomass. So we decided to isolate and explore just the parrotfish biomass. Unlike the US Virgin Islands sites though, where we saw almost a distinct separation of the parrotfish biomass above and below 30 meters, we did not see that separation here. Considering that this data set had fewer number of sampling years, that trend may exist, but we are unable to discern it. However, we are able to see that the average parrotfish biomass across the sampled years in Puerto Rico is less than that of the US Virgin Islands, with the average parrotfish biomass in Puerto Rico being between 0 and 20 grams per meter squared, while the average parrotfish biomass across the sampled years in the US Virgin Islands, if you recall, was generally between 0 and 40 grams per meter squared. To reiterate, the study by Shantz et al. 2020 found that the average parrotfish biomass was approximately 12 grams per meter squared in the Bahamas, approximately 19 grams per meter squared in Jamaica, and approximately 24 grams per meter squared in Turks and Caicos. And to bring it all together and back to our hypothesis that sites with higher herbivore biomass would show greater change in coral cover over time, we decided to run a linear regression to determine if this relationship was present between the two. We did not find a significant relationship between total herbivore biomass and the change of coral cover over time for either the US Virgin Islands or Puerto Rico. However, since we noted a possible trend in the total herbivore biomass and depth with the US Virgin Islands sites, we decided to separate the sites between the shallow sites, which we categorize as less than 30 meters, and mesophotic sites. On this figure, which compares total herbivore biomass to the change in coral cover in the US Virgin Islands, the x-axis is the average or mean total herbivore biomass in grams per meter squared. And the y-axis is the change in coral cover over time, with anything above 0 representing a growth in coral cover and anything below 0 representing a decline in coral cover. Shallow sites, which are less than 30 meters, are represented by the light blue diamonds, and the mesophotic sites are represented by the gray diamonds with purple circles. Here we found a weak but significant positive relationship between the change in coral cover and the average total herbivore biomass for the shallow sites. We found no significant relationship for mesophotic sites, nor a significant relationship for shallow or deep Puerto Rico sites. But the significant relationship here with the change in coral cover and the mean total herbivore biomass at shallow sites indicates that depth may be a significant factor influencing herbivore community composition, growth rate of coral community, or possibly algal composition. So given that we found a significant relationship between the total herbivore biomass and the change in coral cover over time in the US Virgin Islands at sites less than 30 meters, as well as the fact that parrotfish comprise a large majority of the herbivore biomass, we decided to see if the relationship held for average parrotfish biomass and the change in coral cover over time at the US Virgin Islands sites less than 30 meters. So on this figure, we have the average scaridae or parrotfish biomass in grams per meter squared on the x-axis, and the change in coral cover over time on the y-axis, with anything above 0 representing a growth in coral cover over time, and anything below 0 being a decline in coral cover over time. With this, we also found a weak, significantly positive relationship, suggesting that parrotfish may play a role in the change and growth of coral cover over time for shallow sites. But with this being a weak relationship, we acknowledge that there are many other variables at play, and that this relationship does not provide a single answer. We would also like to note that the same relationship was tested for Puerto Rico sites, and no significant relationship was found. Since we saw that significant relationship between the parrotfish biomass and the change in coral cover over time and depth in the US Virgin Islands, we wanted to explore that relationship a little bit more. We performed a multivariate analysis to look at how the herbivore communities among different sites were influenced by the presence and abundance of different parrotfish species, and how this related to the change in coral coverage. On this NMDS ordination plot, depth, distance from shore, and change in coral cover are represented through vectors, or the arrows, and show that some parrotfish species are more important to the coral coverage over time than others, as they are closer in proximity on the plot. In the USVI, scarce icerity, striped parrotfish, are very common, but do not appear to be associated with the sites with the positive change in coral cover, as it is not in close proximity to coral coverage on the plot. On the other hand, scarce guacamaya, the rainbow parrotfish, and scarce coeruleus, blue parrotfish, are much rarer species and are associated with positive change in coral cover. This has some interesting implications on the importance of functional diversity in the relation to reef recovery after disturbances. Not included here, we also looked at the abundance of Diadema antillarum, the long-spine sea urchin, which are important grazers, but since the historic die-off, they have not been seen in large numbers. We saw no relationship between change in coral cover and the Diadema abundance. This was most likely due to very low abundance across all sites. Wait, so what does all this mean? Well, let's recap. Our hypothesis was sites with higher herbivore biomass showed faster rates of positive change in coral cover. And what we found was depth plays a role in herbivore biomass and changes in coral cover over time. Parrotfish are important players and may influence changes in coral cover over time. And some significant relationships were observed in the US Virgin Islands, but not observed in Puerto Rico. Therefore, more data are needed to explore possible confounding factors.
And we still have our work cut out for us. First, we are wanting to explore if coral species composition plays a role in the variation of reef recovery across sites and data sets. We need to further examine species composition because different corals have different growth rates and could therefore impact the amount of coral cover over time. For example, Acropora palmata has a much faster growth rate than Orbicella species, which could influence how quickly these species recover from disturbances. This shows that species-specific growth rates could influence how an entire reef recovers from a disturbance, regardless of the amount of grazing or herbivory that's happening. And second, we want to look into the size frequency distribution of parrotfish, as well as their functional roles. With our results indicating that there is a relationship between herbivore biomass, in particular parrotfish biomass, and the recovery of coral reefs after disturbances, we cannot neglect that differences in the functional diversity of the parrotfish community may also play a role. Parrotfish feeding mechanisms, for example, scrapers, grazers, and excavators and bioeroders that we talked about earlier, differ among species, and this can result in each species consuming different types and amounts of algae. Here we depict the amount of macroalgae eaten on a reef by various species, and we can see that queen parrotfish exhibit more control of macroalgae by scraping than other species do as excavators or bioeroders. Furthermore, the size, age composition of these represented species can also influence their impact of controlling macroalgae on reefs. Juveniles of the various species and functional groups consume different amounts of algae, both in relation to their adult counterparts and other species, which further adds to the species-specific influences that parrotfish can have in managing macroalgae and ultimately impacting reef recovery. And so knowing that foraging rates may vary between species and size classes, we would be interested to investigate the composition of parrotfish species, as well as the size classes within species at our sample sites to gain a better understanding of how these differences may influence coral recovery rates that we observed at our sites. And before we close, we would like to extend our sincerest thanks to everyone involved in our work this semester. First and foremost, we'd like to thank Dr. Marilyn Brandt and Dr. Tyler Smith for leading us in this journey and answering all of our questions, and for taking time to be awesome. The UVI core faculty for all of their input and guidance on the various components of this project, the TCRIP PIs and PRCRIP PIs for allowing us to use their data and for being available throughout the process for questions and advice, Miguel Hernandez for introducing us to the PRCRIP data and the MBON network, Razi Ennis and Sean Cattison, both TCRIP research techs, for being available to us for questions about all the data things. And lastly, this capstone and webinar could not have happened were it not for the awesome work by all of the 2020 cohort members. Interestingly enough, we were a fully virtual cohort this year, and that presented a number of challenges, but this wonderful group of people were able to get it done. And as you can see by our laughing faces, we tried to keep morale high as best as we could. So thank you again to all of our wonderful cohort members. And with that, we conclude our virtual capstone webinar. Thank you all so much for sparosomaing some of your time and joining us today. We appreciate you all and look forward to answering your questions in just a minute. Remember, you can email your questions to us up to 2 weeks after this webinar at the email listed here. If you have questions now, please put them in the Q&A box on your screen. With that, 2020 cohort out.